Hello everyone, how are you doing? I hope everyone is doing good. So welcome to the video of the same chapter, next video of the same chapter, that is reproduction in plants. In the previous video, we covered about the different parts of the flower and what is the function in detail. Now we are going to take one part of the flower and go into much more details into it, and we are going to discuss about how the gametes are produced. So there are two types of gametes, the male and the female, that we have to cover. In this video we are going to cover about the male gamete, which we will study under the heading of microsporogenesis. Before that, we are going to quickly revise and uh, uh, see the structure of anther in more detail. So let's begin with the structure of anther. Okay. So in the previous video, we covered about the filament and the anther. Okay. So the filament and the anther together, they are known as stamen. The stamen is the male reproductive part of the flower, which is also known as androecium. Yes. Now we dissect and go deep into the structure of the anther, because this is the place where the male gametes will be developed. Okay, where the male gametes will be developed. So you can see, if you do a cross section of the anther, this is how it will look like. आप अगर इसको cut करोगे, it will look something like this. And this is the stalk, the filament that is coming over here. So let's begin. Anther, it is the topmost part of the stamen. Yes, so it is terminal, and most of the anther is bilobe. Bilobe means, you can see over here, एक lobe and दूसरा lobe. Okay, like this, one and one another. Like you can take an example from the black gram or the white chickpeas. So it has two parts, two lobes. So when you break open it, you will get two parts, two lobes. Okay, that is what we mean by bi. Bi stands for two. Okay, again, each lobe will have two parts in it. It will have two thecae in it. Okay, so like this, if it is bilobe over here, उसमें भी internally they will have another two sections. So in all, you will have four sections in a particular anther. Okay, so that is what I mean by four stands for tetragonal. Okay, tetrad or tetragonal. It's a tetragonal structure consisting of four structures. Now, what is this four structures? These are known as microsporangia. You can see these circular structure 1, 2, 3, and 4. Okay, outside, from outside, it will be bilobe structure. Okay, when you cut it, you will see four structures in it. Okay, four corners located, these microsporangia, something known as microsporangia, at four corners. Okay, so in this case, you will call them as a tetrad or a tetragonal structure. Micro means very small, minute. What is sporangia? Remember in 11th standard we were discussing a lot about the spores, asexual spores, sexual spores. These are structures, these are granular structures. Here it is standing for the male gamete. Very minute male gametes. Small granular structure. Hence the name microsporangia. Microsporangia is the sac. जहाँ पे ये मेल गैमीट्स बनेंगे, जहाँ पे ये मेल गैमीट्स बनेंगे. So it is the sac, it is the place, it is the location where the male gametes will be produced. Okay, so each lobe will have two microsporangia, as we have just discussed. Two lobes, each lobe having two microsporangia. So in all, 2 plus 2 will give you four lobes of four parts or four microsporangia. Okay, a tetrad structure. And what is this microsporangia? It is nothing but the sac, a place where the pollen grains will develop. So we will call it as microsporangia developing into a pollen sac. And this is a place where these male gametes will be produced. Where the male gametes will be produced. Okay. So where is the male gamete getting produced? In the microsporangia. Where is the microsporangia located? In the anther. Okay. What is the anther? It is sitting onto the filament. It is the male reproductive part. Together the anther and the filament, it is known as stamen. That is the androecium. They serve as the essential floral part of the flower. Okay, so this is the structure that you need to know. In the exam, they might also ask you about the TS of anther that we're going to do in detail. Okay, so that was the overview of the structure of anther. Now we go into deep details of the anther. So up till now, we just had a look on the outer structure. Now we are going deep over here. Deep over here. What are these layers? Okay, that we will discuss. Right? Okay. So let's begin with the details of the microsporangia. Okay, you can see the labelings. Let's just quickly first see the name the labels and then we will discuss in detail. So the very outermost layer, epidermis. Okay, epidermis, like you have the skin, which is the outermost layer of your body. We call them as epidermis. Then you have the mesoderm, epiderm, mesoderm, endoderm. Similarly, the very outermost layer is epiderm. Underlining layer, uske niche wala, inner to epidermis, endothecium. Then moving more inside, so we are coming from outside to inside, step by step, inside. Like you open one door, go into another room, then you open another door, you move into another room. So you are going deep into this. Then these layers, middle layers. Then finally, these red structure, red structures you see, these are the mother cells. We call them as. So you might be surprised that we are discussing about anther. Anther is the male reproductive part. It produces the male gametes. And why do we call them as mother cells? Microspore mother cells, MMC. Actually, it's like generally whenever there is parent cell giving rise to dividing and forming daughter cells. Generally, parent cell dividing and giving rise to daughter cell. Okay, so usually parent cell ko we refer to as the mother cell. Okay, that is why here we are calling them as microspore mother cell. And in when we will discuss about the female reproductive part, there we will call them as megaspore mother cell. Remember, microsporophyll and megasporophyll. So here pollen grains are very minute structures. That is why they are known as microspore mother cells. Then you have this tissue over here, underlying cell around the microspore mother cell. This particular section is known as tapetum. Let's see all these structures in detail, beginning with the microsporangium. What are the layers? So 4 layers, very important. 4 walls. Number 1, epidermis. I just put it number 1. Endothecium, number 2. Middle layers, number 3. And then the layer of tapetum. This is the cell which will actually divide into the daughter cells, that is the actual pollen grains. But the wall, the structures, the cellular structures which is there from epidermis, endodermis, middle layer, and the tapetum. And in there is the mother cell which will divide and give rise to pollen grains. Okay, so microsporangium is surrounded by these 4 layers known as epidermis, endothecium, middle layers, and the tapetum. Okay, the outer 3 walls is for protection. The outer 3 layers is for protection and for dehiscence of anther. It will help in dehiscence of anther. Dehiscence means what? When the cells have been divided, pollen grains are formed. Now, when they are ready, so for burst opening, okay, for burst opening of the anther, it will help in this process of burst opening of the anther to release the pollen grains. So here these cells will help. Okay. So the outer three layers will protect as well as they will help in the dehiscence of anther. Clear? Then the tapetum, which is the innermost layer, you can see after the middle layer, this layer is the tapetum layer. So tapetum will help in the nourishment of the developing pollen grains. So the parent cell, when it undergoes mitotic cell division, daughter cells are formed. So for all this, nourishment is required, nutrients are required. That will be provided by the tapetum, the layer which is known as tapetum. So nourishment will be provided by the tapetum. Okay. So cells of the tapetum possesses dense cytoplasm, and that is why you can see some dots over here. And generally, they can have more than one nucleus. What is the understanding that usually one cell having one nucleus, but this is a specific characteristic of the tapetum cells that they might have more than one nucleus. Okay, so these tapetum will surround the microspore mother cells, which will give rise to the pollen grains. So microsporangia made up of 4 layers: epidermis, endothelium, middle layers, and tapetum. These are just protective layers. The very first protective layer, epidermis. After that, endothelium. After that, protective layer, middle layers. After that, the nourishment giving cells known as tapetum, and inside that is the microspore mother cells, which will finally divide and multiply and give rise to the male gamete, that is the pollen grains. And the 3 layers, which is the epidermis, endothelium, and middle layers, together they will act as protection, and also they will help in dehiscence of the anther. Means before maturation they will not let the anther to burst out. So only when the pollen grains are ready, they are matured, and when they are ready to be released out in the environment after the division and maturation development has taken place, then only they will help in burst opening of the anther. Till then, it will not allow these layers, these all 3 layers will not burst open and release the pollen grains from the anther. So basically, they are helping in the dehiscence of the anther. Both ways. Clear? Make a note of it. Any word or any line you don't understand, make a note of it and you can ask in the live class. Okay, so this is how the structure will look like. You might be asked in the exam to draw the structure or the transverse section of the anther. So you need to know this structure. Basically, drawing is not very difficult in this diagram. It's just the cells that you have to draw in layer in concentric circles. The labeling is very important. So labeling you have to remember clearly. So this is just a cross section of how the layers are there. This is how a tetrad of microsporangia looks like. Okay. So now moving to the microsporogenesis part, having discussed about what is microsporangia, where the pollen grains will develop. Now we discuss about how these pollen grains are formed. Mother cells से daughter cells कैसे बनेंगे? So this process is known as genesis, means formation, making of what? Microspores. What are microspores? Nothing but the daughter cells or the pollen grains or the male gametes. These are all interchangeable. You can see over here. Let's just discuss the diagram. This is the flower. We are into the anther, moving more inside the anther. That is the microsporangia. Here we have 1, 2, 3, 4 parts, 4 lobes. Let's just take one microsporangia. Microsporangium, microsporangium for singular, microsporangia for plural. Okay, so this is the pollen sac. This is the mother cell. That is why the chromosome state is diploid. It is what? Diploid. 2N. Parent cell will always be in the diploid condition. What is the name over here? Microsporocyte. Now this microsporocyte will undergo cell division. What kind of cell division? What kind of cell division? It will undergo meiotic cell division. Why? Because it has to produce gametes. Remember, two types of cell division are there. Whenever we discuss about reproduction, reproductive cells, formation of gametes, which type of cell division will take place? Meiotic kind of cell division will take place. So microsporocyte, that's the mother cell, diploid condition mein, it will undergo cell division. And what is the outcome of the meiotic cell division? 4 haploid daughter cells are formed. So you can see over here, 4 haploid daughter cells are formed. Haploid means half the number of chromosome from the parent cell. So parent cell, if it is what if it was diploid, uska आधा क्या हो गया? N हो गया. Okay. So 4 microspores will be formed. Yes. So these further microspores will individually develop and they will now multiply and increase their number by which type of division? Mitotic division. 4 microspore बन गया तो क्या 4 pollen grain से काम बन जाएगा? No, there has to be more number of pollen grains, okay, for the pollination to take place. So these more number of pollen grains, how will they be formed? By mitotic division. So once the meiotic division takes place, now these cells, microspores which are formed, they will now undergo rapid multiplication and division. Now everything will remain the same. The chromosome number, everything will remain the same, because now what type of division is happening? Simple mitotic division. Okay. And then the pollen grain will be formed. So this is how the pollen grain will look like. Okay. Let's just revise it again. So the sporogenous tissue transforms into pollen mother cell. Okay. What is this pollen mother cell? That's the microspore mother cell or the pollen mother cell. This will undergo meiotic division to form what? Microspores. Okay, the daughter cells, the 4 daughter cells, specifically we here call it as microspores. What will be their chromosome condition? Half the number of chromosomes, that is N, haploid. Okay, this whole process from year to year, okay, this process is known as microsporogenesis. What it is known as? Microsporogenesis. And finally, these microspores are nothing but they will develop into the pollen grains. Clear? Take a note of it. Ask any doubt if you have. Make a note of it and then you can ask about it. So that was all in this video. We had covered about the structure of anther in detail. This is one of the most commonly asked diagram in the exam. So practice drawing it and remember the labelings of the PS of anther. And then what is microsporogenesis? That is, now we understood the structure of the microsporangia and how this then we moved on to how the pollen grains will be developed from the mother cells. So mother cells will be 2N, they will undergo meiotic division, result into 4 microspore haploid condition. Then they will undergo mitotic division just to increase their number. Okay, so in the next video we will cover about, now we know how the pollen grain is formed. So we will discuss in detail about the structure of the pollen grain and how this pollen grain will further develop and go and fertilize the female egg, female reproductive part. So that we will see in the next video. Thank you all of you. Stay tuned and we will see you. I'll meet you in the next video. Okay, take care. Bye. See you all.